Hello students, welcome to Civil Service Gurukul. We are back again with another Science and Tech section for upcoming UPSC Prelims 2025. So we have already covered space, defense, medicine. So today we are taking up a difficult theme, biotechnology. Because every year we see the most difficult section to crack from science and tech continues to be biotechnology because the level of questions being asked in this section is slightly on higher side. It is the only topic which is mentioned in your plus one, plus two biology textbook. So that's the reason why UPSC experiments with this theme on quite, you could say, deeper level. So the three topics which I am taking up in this particular lecture are related to some positive and controversial themes. Firstly, we will be decoding the different types of organ tissue transplantations. Then we will shift to the most happening topic of gene editing experiments. And finally, we will see which GM crops made headlines for right or wrong reasons. So let's start with the first theme for today's lecture, the organ or tissue transplantation. So whenever there is an emergency or some, you could say, mishappening with some individual, at times we need organ or tissue donors. So ideally, there are 4 different permutation combinations which are available with health authorities, with which they try to experiment in order to save someone's precious life. So the 4 permutation or combinations in the transplantation or grafting part are, as you could see on the screen, autograft, isograph, allograft, and xenograft. So basically, while preparing this topic, you should have a crystal clear clarity in the conceptual part what exactly these 4 terms denote. The first term has the highest success rate, it's the least controversial, and it's the most preferred type of transplant in case of any individual. The term autograft, as you could see on your screen, refers to when transplant takes place from one part of the body or organ to another part. So basically, if someone is having a burn injury or having some major accident through which you could say there has been facial disfigurement, at times we might just borrow or you could say transplant a tissue or a structure skin from one invisible part of the body to the visible part. So basically this is your own body structure or part, so there is the least possibility of body being rejecting this part. So immunologically also this is a most preferred one. So typically you could say skin, hair, bone are the common transplant which are often carried out by in this domain, and they continue to be a popular choice in healthcare facilities. Coming to the second type of grafting, the isograft refers to the transplantation or grafting taking place between 2 identical twins. Since they are identical twins, they have more or less similar morphological or phenotypic characters, and also they have almost similar genetic makeup. So there is a high probability of transplant being successful in this scenario as well. The only limitation of this particular transplant is that identical twins are a rare occurrence in a common population. Generally, in 10,000, you could say there are 1 or 2 groups which can be referred to identical twins. So basically, there is a logistic challenge while you could say referring to this identical twin part. Otherwise, we can say it also has a similar success rate, and it is more or less a preferable mode as well. Now, coming to the third major grafting, it refers to the allograft, and this one is the graft which is often in use for sometimes controversial reason as well, because lot of private hospitals at times carry out such transplant in nefarious way. Susceptible donors are misled at times. Their kidneys are transplanted to save some rich man's life, and no proper, you could say, the consent or you could say matching immunological are done, which reflects the sorry state of governance in healthcare facilities. But when they are carried out in an ethical, in proper, regulated way, they can save someone's life, because at times, even though human beings have 2 kidneys and 1 is required, but at times when both fail, especially after you could say dialysis, this is the only you could say life saved. Also, some you could say difficult transplants like in heart, lung, liver are also carried out when person is clinically dead and it has given a consent that after his death he is ready to donate organs. Then this transplant could be quite handy in saving multiple human lives, especially if the person happens to die at young age. So even though in India there are a lot of rumor mongering and a lot of people are quite skeptic about donating their organs or you could say tissues after clinical death, but still we could say India is seeing a progressive trend in current decade, but more can be done on awareness part. So basically this transplant carries a little you could say moderate success ratio, because at times when you are transplanting some organ or a tissue from one human being to another, immunological reaction at times could be the reason that this transplant might not be as successful as was ISO or autograft. So we have to be little careful while taking this bold decision of transplanting. So in a nutshell, we can say this is a transplant in which the transplants are carried out between two different individuals, but they belong to same species. Coming to the final transplant is the one which is the most controversial one, and it's the one which made headlines in last, you could say 2024-2025 timeline, is the xenograft. It is ornamentally called xenotransplantation. Now, in this transplantation, when there is a huge emergency, we try to cross species barrier to save human life. In this, typically a mammal is sacrificed and its organ is transplanted to a human being. Even though this mechanism is quite controversial in nature, because you do not take the consent and then the animal has to be sacrificed majority of the time for this bold experiment, and then you have to rely on heavy genetic engineering so that the transplant is a successful one and body does not immediately reject, because these are the most riskiest transplants which are carried out and they carry a poor success ratio. So we can say now you have a clear idea of what are the four types of transplants possible. In to summarize, auto means same body, iso means between identical twins, allo means between same species, and xeno means between two different species. But these species have to be close, you could say, to family. You cannot just say I will transplant a fish to a human body. At least the mammal group is to be used when you are carrying out such bold endeavors. Now, coming to why we are taking up or focusing on this transplantation part so much in this particular, you could say, prelim based series, because in last 2 years there have been two bold experiments which got mixed success results. The first one catered to a person in which a pig heart was successfully transplanted, and it was an ideal example and a breakthrough for xenotransplantation, even though within a month the person died, and there was a lot of hue and cry that why this such controversial experiment was carried out in first place. But nevertheless, it opened a new Pandora box in which in emergency or some critical situation when there is no reasonable donor available from human side, we can experiment with this last resort method, because at least it will prolong someone's journey by few weeks or months at least. So we are at advanced stage of, you could say, medical evolution, where even we are trying to cross species barrier. Now the other one was also quite successful one, was in which once again they chose pig and transplanted its kidney to a human being, and it also successfully worked for a couple of months. So we can say this is a new theme which might not be a norm as of now, but there is no harm in experimenting with such technology with the right intentions. And we should have a proper checks and balances and regulatory mechanism so that do's and don'ts are not crossed often and they are only resorted when the situation or healthcare demands. So in this particular case, as you could see on your screen, the xenografting technology has been briefly demonstrated. A pig which was genetically engineered gets sacrificed its body organ, like in this case liver and heart they have showcased, are tried to successfully imbibe to human body. So this is a experiment which has quite potential, but body, since you are trying to cross a species barrier, it does not carry the same success ratio as of now. Coming to the second major theme, which is quite favorite of UPSC in last, you could say, 5 years. Already 3 questions have been asked from this section. There was a question on CRISPR, you could say 3 or 4 years back. Then it was followed by what exactly is Cas9 system related to. Then they had asked about the applications. What exactly are the basic applications of evolving genetic editing experiments? So in this topic, first of all, you should be clear about what is the enzyme-based system which is making headlines and making this technology possible. So it is none other than CRISPR-Cas9. So basically, this is a enzyme which was isolated from, you could say, bacteria or a prokaryote, and it has unique cutting abilities. It can specifically cut DNA segments at specific site, and these could be transplanted or edited at will. So you can say they are molecular seeders of a kind which are making technical, or you could say long lasting genetic permanent changes in our DNA makeup with some promising results. And the two broad types of gene editing which are making headlines currently are the DNA editing and the RNA editing. So you should be able to, first of all, to start this topic, have a clear difference between what are the two basic nucleic acids like DNA, RNA, and what are the basic differences between them. To just have a short summary of the differences, I am just having a quick word. The DNA has deoxyribose as sugar, whereas RNA has ribose as sugar in its structure. Then the nitrogen bases in DNA are typically the adenine, thymine, cytosine, and guanine, which we call ATCG. Whereas in case of RNA, the nitrogen bases are adenine, uracil, cytosine, and guanine. Now, DNA is mainly present in nucleus, whereas RNA is found both in nucleus and cytoplasm, and it is of multiple types like T, M, or rRNA. So once you have decoded the basic general science part of this nucleic acids, then only you can advance to what exactly is DNA editing or RNA editing. In DNA editing, you are using CRISPR-Cas9 enzyme-based system isolated from bacteria to make specific changes in the existing species without involving some other different. So the good thing about the gene editing experiments are they are not crossing the species barrier from one species to another. They are just editing the existing genetic makeup to its full potential, or you could say the right potential, just eliminating disease or defective parts. So basically you can say it's the advanced version of gene therapy experiments. In RNA editing, the changes are of transient nature, they are not permanent, and they are quite safer to carry out, and they do not have the long lasting effect. So basically in this year we have seen the emergence of RNA editing technology in which ADAR enzyme based system, once again isolated from bacteria, is making headlines. Even though the full form is quite technical one, no need to mug up adenosine deaminase, you could say, acting on RNA. But you should have idea that these are two buzzwords in the biotech sector currently. Now, you should also have a fair idea from Bain's perspective, what are the potential applications. The basic applications scattered to gene editing refers to the treatment of genetic disorders. Now, genetic disorders are inherited disorders which are passed from one generation to another. For example, we can say there is a person was a color blind, a person, and then this defective gene was passed on from grandparents to parents, then to offspring. So in such you could say genetic disorder, there is possibility we might edit the defective part or remove it and then try to edit the genes to its functional state. So definitely this technology has a promising future in treating, you could say, genetic inherited disorders, which could be the idealism goal of eugenics or euthanics. Then coming to immunotherapies, some unconventional diseases which continue, you could say, be a malady for a lot of population like cancer. Some new type of treatments can be evolved using these breakthrough, you could say, editing technologies. We might be able to, you could say, modify our immune cell to fight these cancer cells for better. So there is no harm in experimenting with such new treatments. Also, this technology can be quite handy in discovering new vaccine or drugs at faster pace, because we see after the COVID pandemic, we should be alert and at vigil, and whenever some new disease outbreaks, we should have some new treatment protocols to handle them urgently. And finally, this technology is not just limited to healthcare. You can also use it for editing crops. And already there have been a lot of crops which are edited to produce higher yields. And unlike the GM crops in which you could say transfer genes from one species to another, these are comparatively safer, even though they continue to face the ethical barrier from the community. And the difficult part, or the you could say the controversies or cons of this technologies revolve around that this novel technology has a potential to be misused for bioweapons. You might just modify a person for some nefarious mutated means. There is a possibility that you might reduce the genetic diversity as well, because you tend to just promote the best or the idealized version of homogeneity, and heterogeneity or diversity or a defective one might get reduced. Also, the powerful countries will get more powerful by producing a superior race, replicating superior genetic traits, whereas some poor countries which are already malnourished and are not able to compete, they might not be able to afford this breakthrough technology at faster pace. So basically, it can create a divide in society, and the most important one which limits its use in human beings continues to be that it's a new emerging technology. We need to develop proper protocols and regulatory mechanisms. Only then we should give it a go ahead. So we can say the safety challenges continue to be, because when you are just modifying DNA or RNA at such a microscopic level, there might not be immediate ramification, and after a few decades or generation, the real, you could say, mutation or some harmful effects might be visible. So it's a lot better to have a cautionary principle rather than to feel sorry at the immediate acceptance. So we can say this technology is continuously evolving, but you should have a clear idea about what exactly are the different types of gene editing experiments which are currently making headlines. Coming to the final part, it's once again in prelims, it's quite an important one. The GM crops which makes headlines. So this year, the golden rice made headlines for the wrong reasons, because last year we had seen Philippines was the first country in developing nations to finally give it a go ahead for commercial planting. But within, you could say, a year, it has decided to reverse its stand, and once again, it is a setback for GM crop proponents, because it has put the trials on hold once again. So when such a topic is in news, they might not ask which countries have pulled it back or which are accepting. They will just ask what exactly is golden rice. So golden rice, as you could see on your screen, is more or less a normal rice, but which is having a golden hue in its color. So it's not just that they have tried to just make it more aesthetic, because white rice or brown rice are equally aesthetic, or there is not much barrier that someone is consuming them. Their real motivation to produce this golden rice was it incorporated beta carotene segment. So beta carotene is precursor for vitamin A, and from there this topic becomes important from pill. So make sure you are clear what exactly is the role of vitamin A in human body. It's required for healthy skin as well as normal eyesight, because the scientific name of vitamin A, retinol, signifies that it is responsible for producing rhodopsin pigment in our rod cells, which are primarily helpful in when we are seeing dim light, or you could say at night time. So person who is having vitamin A deficiency is likely to suffer either from night blindness or from xerophthalmia or toad skin. So basically the intention of the development of golden rice was straightforward. They not only wanted that this particular rice provide a rich starch or carbohydrate content, it will also supplement with vitamin A content. So a population where you feel the vitamin A deficiency is prevalent, there was no harm in experimenting with proper checks and balances to allow this crop to be fed. So basically, because this vitamin A deficiency is more pronounced in developing countries, so that's why the acceptance of developing world is important for to make this product and important. So once again, you can see that this GM tech crop has not finding the takers which it intended to focus because of the ethical controversy. Other GM crops which made headlines were once again the BT cotton, HT cotton. The BT stands for Bacillus thuringiensis. It's a pest-resistant cotton variety which provides protection against the pink bollworm. Even though at isolated spots, we continue to see other pests like whitefly damaging cotton crops. So currently it's a mixed bag. And then HT cotton is herbicide tolerant cotton, which is once again an important crop. Even though India has not allowed any food crop as of now, but in developed world, a lot of other food crops continue to make headlines, right from BT brinjal, BT corn, GM potato, GM tomato. So make sure you have fair idea about what are the specific characteristics these particular crops carry, so that once a match the following or incorrect pair question comes, you are having a thorough background or understanding of the themes. So this is what we had in today's, you could say, biotech update series in our prelims cruise program, in which we are mainly catering to a quick revision or having some basic foundation laid for the important topics for upcoming prelims. If you are having any difficulty in understanding, if you feel that pace was too much or you are not able to grasp a specific theme from this lecture because it's a difficult theme, feel free to connect. You could either visit Civil Service Gurukul or you could drop a query in comment section. We will try to get back to you as soon as possible. So once again, best of luck to all aspirants who are appearing in prelims. Only few days are left. Make sure you stay focused and just continue to practice, you could say, multiple mock tests to get the confidence. Have a nice day.